Hello and welcome to the A22 review video for cell structure. Pretty sure it'll focus. There we go. Alright, so what did we learn about in cell structure? Standard one. So standard one says that cells are the basic structural unit of all living things. So another way of saying that is cells are the smallest unit of life. Therefore, all living things must be made of cells. This kind of comes with the cell theory, and so it's part of the nature of science, is the cell theory. And so the cell theory says three things. Cells are the smallest unit of life. All cells come from pre-existing cells. And all living things are made of cells. So they're kind of redundant, but what this means is it tells us to predict that basically if we find something that's living, we know it's made of cells. And so whenever we discover something that's living, we never stop to think if it's made of cells. We know it's made of cells. It also means where do we put viruses, right? We'll talk about them in another unit, but the viruses obviously are not made out of cells, not in our traditional sense of the word. All right, standard two. Standard two is, can you use a microscope? Okay, can you use a microscope? And so you're supposed to be able to, mount things onto slides, measure sizes, etc. So you have your coarse focus. That's gonna be the big knob, and that's gonna, coarsely focus. And so that's like big changes in the focus. Then you have your fine focus, which is going to be the smaller knob, and that is going to be tiny changes. You can stain tissues, right? So there's a bunch of different stains, and the different stains are for different molecules. So depending on the stain you use, you'll see different molecules. So we used like 3 different stains and looked at the different molecules. So then we can calculate its aim. So you can calculate the actual size, the image size, and the magnification. And so it's one of those like triangles that looks like this. Except I am now thinking I did it wrong. Yeah, I did. Whoops. It's okay, we all make mistakes. As soon as I looked at it, I was like, that's not what the formula is. There's the formula. Okay, and so what you can do is, if you know the actual size and the image size, you can divide those two to get the magnification. Or if you know the magnification, you can calculate the other two. So how do we get the image size? Image size, you use a ruler. You pretty much always want to measure in millimeters, and I'll explain why in a second. For actual size, we typically get this from a scale bar, or you could be asked to calculate it. But it often comes from a scale bar. It's typically in either micrometers or rarely nanometers. The reason we're going to do millimeters is because it works like this. You have meters, millimeters, micrometers, and nanometers, and they're all 1,000 apart. And so if you have something in meters and you want it in millimeters, you would divide by 1,000. So depending on which way you would go, you would either multiply or divide by 1,000. And so if you wanted to go in this direction, you would divide by, you had meters and you wanted to go this way, you would multiply by 1,000. And if you had this and you wanted to go this way, you would divide by 1,000 each time. So like between this level is times 1,000. Between this level is times 1,000. And between this level is times 1,000. The reason why is because of what these things mean. So millimeters means times 10 to the negative third, micrometers means times 10 to the negative sixth, and nanometers means times 10 to the negative ninth meters. Right? So that is our conversion factor. You have to divide these. They have to be in the same units. They have to be in the same units. If they're not in the same units, you can't divide them, right? That would be a big no-no. Can't do that. Let's see what else. Scale bar, taking photographs. Okay, that's pretty much it for this standard, I think. 3, development in microscopy. So this one is an interesting one. This is a newer standard. So besides our normal light microscope, which is what we use in classroom, you have all of these different ones. And so we'll start with, there are fluorescent stains for microscopes, for light microscopes. And these fluorescent ones, they need a special little add-on. But basically it allows you to stain it with these special little things that fluoresce. Okay? And so what you've got going on is they fluoresce and they light up. And what this allows you to do is just see your target molecules. And so it helps you narrow in on like the specific thing you're looking for. In a light microscope, you can see everything still, but in this one it's dark and you can only see the things that like fluoresce. Building off of this, it's similar is immunofluorescence. Immunofluorescence is going to be more targeted because you're using antibodies. So we haven't really learned about antibodies yet, but we will learn about them next year. And they're basically attaching to proteins, but they're going to attach to specific proteins. So that makes this one very good for looking for specific things. That's next. Oh, electron microscope. So our electron microscope, right, it allows us to see really, really small things. It's got that 1 nanometer resolution compared to the light microscope, which is about 400 to 700 nanometers, the wavelength of light. And so we can see things that are a lot smaller with electron microscope. We can also use tomography to get 3D images. So tomography gives us 3D images. Freeze fracture allows us to see inside like membranes and to see what the membrane looks like. That's how we figured out about the structure of the membrane was done through freeze fracturing. And then so this is like it's basically, it sounds like you freeze it, fracture it, and it allows you to see inside of the things a little easier. And then what's the last one? Oh, our cryogenic is like the newest technology. Our cryogenic electron microscopes, these preserve the samples. And so the other two destroy them. So in these two they're destroyed, and in this one they're preserved, which allows you to reuse the sample again. And this one tends to use like X-rays to get it to do it without destroying it completely. All right, microscopes. 4. What do all cells have? All cells have DNA, cytoplasm of water. Oh, look at that, it focused on its own. So proud of it. Cytoplasm of water, and then plasma membrane, that's our phospholipid bilayer. Made of lipids. So that is found in every living thing. So this, right, is our genetic material. This is water is the medium for all life. We did a whole unit on that. And then the plasma membrane creates that barrier to separate it. Prokaryotic cells. Prokaryotic cells have a cell wall made out of peptidoglycan. Some of them have a thicker cell wall than others, right? So gram positive and gram negative refers to how thick that cell wall is, but they all have one. It's just whether it's how thick it is. They have a plasma membrane, right? Duh, that was in the last one. I guess I'll write it anyway. Plasma membrane. They have DNA, but they have special DNA. It's naked, and it's found in a loop. Okay, so it's not linear, it's in a loop. Yeah, but that loop doesn't have to necessarily be a perfect circle. That's fine. 70S ribosomes. That refers to the size. So they're different ribosomes than in eukaryotes. These are in prokaryotes only. Those are the main things that they always have. They can have things like flagella, but they don't have to have a flagella. They can have pili, but they don't have to have pili either. Eukaryotic cell structure. You need to know how to draw it, but we did the drawing separately. Alright, eukaryotic, or you should be able to draw it probably. Eukaryote cell structure. So these are different, right? And we got to be careful because there are animal, plant, and fungi cells. And so there's a variety of these. We'll talk about that more later. But they have a plasma membrane again. They also have compartmentalization. Compartmentalization. What does that mean? This is a fancy way of saying membrane-bound organelles. Membrane-bound organelles. ADS ribosomes. A nucleus. They have DNA that is linear and bound to histones. What are histones? Histones are proteins. So it is not naked, right? It's attached to protein. This nucleus is double membraned. We'll talk about why in the HL section. And it has pores. The pores are to allow the mRNA to leave. Then you have to have a mitochondria. Every eukaryote has a mitochondria. Multiple mitochondria. Mitochondria is plural. Endoplasmic reticulum. There's two different kinds. You can have both. These are all about like packaging lipids and proteins. Golgi apparatus. This is for exocytosis or for making things for the membrane. And then vesicles and vacuoles. They're all, vesicles and vacuoles are like single membrane. Oh god, I spelled single wrong. Single membrane. A lot of these others are double membrane. So the mitochondria has a double membrane, nucleus has a double membrane, chloroplast has a double membrane. But most of our vesicles, like lysosomes, are single membranes. So let's see. They also have a cytoskeleton, and the cytoskeleton helps keep this cell structure, right? And so cytoskeleton is made out of microtubules and then microfilaments. All right, and that's our eukaryotic cell structure. Processes of life. Now we just go, whoa, processes or functions of life. What do we got going on? So, homeostasis. Maintaining internal balance. So if you're in HL, we just saw that in another unit. Maintaining internal balance. With the contractile vacuole. All right, homeostasis, metabolism. Metabolism is like how it's our enzyme catalyzed reactions. There's protein synthesis, cell respiration, photosynthesis. Those are all part of metabolism. Nutrition, it must acquire energy. This is our autotroph versus heterotroph. Movement, it needs to be able to move. I'm not going to write anything else for that. Growth, I think we know what growth means. Excretion, excretion is removal of waste. Response to stimuli. It's like if someone turns the lights off, you like go, ah. And then reproduction. Response to light is honestly one of the most common ones. Then reproduction, that can be sexual or asexual. And so they have different versions of this. But they have different versions of this, but they all have some version of these different things. All right, standard 8. Oh, this is our animals versus plants versus fungi. Animals, plants, and fungi. And so for this one, we're talking about what are the differences. And remember, when we do a T-chart, you need to specifically say that the other ones don't have it. It's not enough to just be like, oh, this one has it. You also need to say the other one doesn't have it. All right, let's start with the cell wall. No cell wall. Cell wall. Cellulose. Cell wall. Chitin. 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 Okay, no plastids. No plastids. This one has the chloroplast and other plastids. Often for like storing starch or pigment. Here we have centrioles. Centrioles. These are used in mitosis. None. No centrioles. No centrioles. Vacuoles, right? They all have vacuoles, but they're small and temporary. Whereas in these, they're large and permanent. Really, now it would have been smarter to write it over to the side like that for all of them. And these can have cilia and flagella. Yes, no, no. Oh, that was way faster. Alright, and then lysosome. Whoops. Lysosome, remember, for digestion with lysozymes inside. Yes, no, no. Okay, great. That is a quick summary of the difference between animal, fungi, and plant cells. Atypical cell structures. Okay, so we have a couple weird ones we need to know. Couple weird ones. So you have aseptate fungal hyphae. Fungal hyphae are like the roots of fungi, right? So fungi spread out, kind of like roots, and that's how they absorb nutrients in. And these aseptate ones do not have no cell partitions. So it's kind of like a bunch of cells merged and became one. Similar concept with striated muscle fibers. So skeletal muscle is striated, and it's striated because it also has no cell partitions, which means it has multiple nuclei in one cell. And so that allows it to make way more proteins. It allows it to make way more proteins, right? Your muscles need a lot of protein, and so it allows it to make more of it. Then we have our phloem sieve tubes, which are living, but they basically don't have anything in them. They have their companion cells that do all of the like things for them, and they're basically just like there. So they also have gaps between them, and they really don't have anything. They don't have a nucleus, they don't have anything else, and so the companion cell does everything for them. Then we have our red blood cells, which also have no nucleus. They're basically just filled with hemoglobin, and they float around, and then they die. So that's standard number 9. Those are the exceptions to cell theory. Or atypical cell structure, excuse me. Alright, 10 and 11 is the drawings. And I'm just gonna let you look those up, right? Could be its own whole video. So standards 10 and 11 are the last SL standards, and these are the drawings / pictures under a microscope. And I am not a microscope, so I can't make those appear. So that is the end of the SL. From here, we have our HL standards. There are just 3 of them. Standard 12 is endosymbiosis. Endosymbiosis. How did eukaryotic cells come to be? And so it suggests that there was this one cell with a nucleus and reproduced sexually. That's how it started. But it didn't have anything else in it. No really other organelles yet. And so it was chilling. And then it ate a mitochondria. So it engulfed it engulfed a prokaryote. Got ahead of myself. It engulfed a prokaryote and did not digest it. And it formed a mutualism and a symbiotic mutualism at that. I really should have read that in the other order. A symbiotic mutualism. So that's when it's like inside of it. This is very different, right? Most of our ecology mutualisms are not symbiotic at all because that requires it to be like within it. Like it means like literally together. Okay? So this is a different kind. So it didn't digest it, and then this obviously it would outcompete all the rest. And so that then gave rise to all the rest of the eukaryotes. And then this one is weird. So for the chloroplast, so that's the mitochondria. Oh, sorry, that's how the mitochondria came to be. This one, the chloroplast, it happened more than once, but not in all. And it was a cyanobacteria. That's our bacteria that can do photosynthesis, and it was a cyanobacteria. How do we know these things? Well, there's a couple pieces of evidence that they both have 70S ribosomes, right? So everywhere else in your cell you find 80S ribosomes, except inside the mitochondria and the chloroplast. They have a double membrane, the one being the vesicle, the other one being the membrane of the prokaryote. They have naked DNA, just like prokaryotes, and they have their own DNA, so it's double evidence, right? The fact that it's naked means it's prokaryotic, and the fact that they have it is weird because your other organelles don't have DNA at all. And then they have the ability to replicate on their own, and they replicate through binary fission, just like bacteria do. So you can make more of your mitochondria all on their own. Oh, naked DNA, and it's circular, just like prokaryotes, my bad. And then there's one other, they're a similar size to prokaryotes. So there's a lot of evidence that they were once free living prokaryotes that got engulfed and became mitochondria and chloroplasts. So there's really 2 parts to that, right? It's how did it happen, and then the evidence. All right, standard 13. Cells have DNA and genes that code for proteins. And these genes can be turned on or off. When you turn them on or off, this changes the cell. And so different tissues, different genes on. Notice it's not saying there are different genes. Every cell has every gene except for the red blood cells. But it can be changed, and it can be changed by the environment. And by the environment, we mean like your environment as well as like the cell environment. And so this, as the tissue is developing in the embryo, the different cellular environment, the extracellular matrix, can turn genes on or off. And we'll send more. There's a whole section on that, so we'll cover that more later. Multicellularity. Kind of like how the chloroplast has evolved more than once, multicellularity has evolved more than one time, and it's evolved probably about 4 times. And so plants did it, fungi did it, animals did it, and here's where it gets weird, so did algae. Right, so our kelp forest is actually multicellular algae. It's not technically a plant. And so each of these evolved multicellularity independently of each other. And why might it have evolved multiple times? Because it's advantageous. And why is it advantageous? The advantages are larger body size and cell specialization. So those are the 2 big ones. So if you're multicellular, you can have specialized cells, so they can do everything a little bit better, and you can grow larger, which helps you get more resources. And that is A22.